Thank you for the introduction. I am excited to be here today and to talk to you about Cas-CLOVER gene editing in the generation of non-viral off-the-shelf allogeneic CAR T cells. Before we get into the details of how we use Cas-CLOVER in the generation of allogeneic CAR T cells, I wanted to give an overview of Poseida and our innovative proprietary technologies that we use in the development of next-generation cell and gene therapy product candidates. Now, Poseida has a long history of development using both our non-viral piggyback DNA modification system for gene delivery, as well as our Cas-CLOVER site-specific gene editing technologies. These and other tools allow us to support a broad pipeline of cell and gene therapy product candidates with the potential to develop therapies even beyond the immuno-oncology space. At Poseida, we started with the development of autologous CAR T to address both hematological and solid tumors, first for patients with multiple myeloma, which is currently in the clinic, and now with our therapy to treat patients with metastatic castrate-resistant prostate cancer. However, we do believe that the future of CAR T will be allogeneic, and that is something that is reflected in the wide array of product candidates that is currently in preclinical development at Poseida. Beyond the realm of CAR T, we are focusing on the development of liver-directed gene therapies that use both our piggyback DNA modification system as well as AAV to achieve a lasting therapeutic impact through stable gene integration. As you may know, chimeric antigen receptor T cells, or CAR T cells, are a form of cell therapy for the treatment of cancer. And this therapy is based on the transfer of a CAR into T cells, which now allows these T cells to target and destroy cancer cells that are expressing the targeted antigen. Now, these cells can come in two flavors, depending on where the T cells that are used for CAR T manufacture are derived from. If the cells are derived from the cancer patient themselves, we're talking about autologous CAR T, while if the cells are derived from a healthy donor, we're talking about off-the-shelf or allogeneic CAR T. However, not all CAR T products are created equally, and that is not only true for the type of target or CAR design that you're choosing to employ, it is also true for the type of T cell that is comprising your cell therapy product. Now, human T cells differentiate along a unidirectional hierarchy, and at the top of this hierarchy sit T stem cell memory cells, or TSCMs. These TSCMs are long-lived, they're self-renewing, and they have the ability to give rise to all the other T cell subsets that are further down the hierarchy. In contrast, at the bottom of the hierarchy, you can find T effector cells. These T effector cells are fully out-differentiated, and they're extremely efficient at killing the tumor, but they're very short-lived. And it is now believed that TSCMs that are on top of the hierarchy are really the type of T cell that you want to go for in cell therapy applications because they have the ability to act more like a prodrug, which means they're not incredibly efficient at killing the tumor themselves, but they're able to give rise to virtually an unlimited supply of tumor-killing T effector cells that will eradicate the tumor over time. And because of that, at Poseida, we're striving to develop cell therapy products that are comprised of a high percentage of T stem cell memory cells. At Poseida, using PBCMA-101, our autologous CAR T product candidate for the treatment of multiple myeloma, we have learned how beneficial a TSCM phenotype in CAR T can be. We saw that a high TSCM percentage was correlated with best patient responses in the clinic, and we saw a gradual expansion of the cells in patients, which led to significantly increased tolerability, and that led to the FDA allowing us to perform our phase 2 clinical trial in a fully outpatient setting, improving the potential for cost savings and patient access. Lastly, we believe that a TSCM phenotype will be a key to seeing CAR T efficacy in solid tumor indications, as our product candidates have demonstrated unprecedented efficacy in a number of preclinical solid tumor models. However, since autologous CAR T therapies are derived from patient cells, these products have a number of inherent limitations. Because most of the patients are heavily pretreated with other cancer therapies, there is a large variability in both the ability to manufacture the cell products from the patient's cells, as well as in the potencies of the patient-derived cell products. In addition, these products are tailor-made for each patient, which makes them more expensive. We believe that an allogeneic CAR T product made from healthy donor T cells will overcome these limitations. Healthy donor cells promise a higher degree of consistency both in manufacturing and in product characteristics, and generally healthy donors have a higher percentage of TSCM in the pre-manufacturing material. In the allogeneic setting, a single production run will be able to yield hundreds of patient doses, which will dramatically decrease cost, and the product will be readily available off the shelf, which improves patient access. Nevertheless, challenges need to be overcome to create a fully allogeneic CAR T with desirable TSCM characteristics. Efficient gene editing of resting T cells will be required because the more that T cells divide outside of the body, the more they will mature and become terminally differentiated. Further, a high-fidelity gene editing tool, which minimizes the risk of unwanted off-target edits, is required to ensure patient safety. I hope that I can show you today how the Cas-CLOVER gene editing tool allows us to meet these challenges. So how do we use Cas-CLOVER to generate allogeneic CAR T with a high percentage of TSCM? Cas-CLOVER allows us to perform efficient multiplex gene editing in resting T cells, and again, this is crucial for the preservation of the TSCM phenotype. We are targeting the T cell receptor for knockout, which will prevent the CAR T cells from reacting against the host, causing a disease called graft versus host disease, or GVHD. Now, GVHD can be potentially fatal, and so a nearly 100% removal of the TCR in the final product is absolutely crucial. We're also targeting beta-2M, which leads to the removal of MHC class I from the cell surface and aids in the persistence of the product following administration. Cas-CLOVER gene editing, as has been shown in the previous talk, is highly specific, resulting in low to no off-target editing, yielding a final product candidate with a desirable safety profile. In addition to Cas-CLOVER gene editing, we're using a proprietary booster molecule that allows us to significantly increase the production yield of our allogeneic CAR T cells, generating hundreds of patient doses from a single manufacturing run. Using this approach, we are able to generate an allogeneic CAR T product candidate that is functionally equivalent or even superior to an unedited healthy donor CAR T product. Next, let's take a look at Cas-CLOVER's exceptional specificity and why it is so important. In general, all gene editing technologies suffer from the risk of off-target editing events and the associated unwanted mutations that are being introduced into the cell's genome. In a multiplex gene editing system, in which multiple targets are edited at the same time, these mutations can come in two forms, either as a simple off-target mutation that could lead to the loss of gene expression, or in the form of translocations between two editing sites that could lead to disruption of gene regulation or induce aberrant gene expression. Now, we analyze our fully allogeneic product candidate using both an in silico approach and a so-called unbiased approach. The in silico approach has already been discussed in the previous talk, and so I will focus here on the unbiased approach that is based on the guide-seq method. To detect potential off-target sites in an unbiased fashion, the gene editing reaction is carried out in the presence of short DNA duplexes that are incorporated into sites where the DNA is broken, a subset of which may have been caused by the gene editing tool and are therefore potential off-target cut sites. These duplexes can then be used as a priming site to identify the location of duplex incorporation in the genome by a next-generation sequencing. Sites that are identified at a frequency above background or that occur in products from multiple donors are potential off-target sites, and these sites are then surveilled by a next-generation sequencing in the final product candidates. For the interrogation of potential translocation events, we use a next-generation sequencing approach that uses gene-specific primers that bind in the vicinity of an on-target editing site and that allow us to identify the sequence that lies on the other side of the cut site. This approach allows us to identify translocations occurring between two known editing sites, as well as between one known and one unknown translocation partner. Initial data generated using these techniques support the high fidelity of Cas-CLOVER and its low to no off-target editing capabilities. We have seen the expected translocation events between the TCR and the beta-2M cut site at a very low frequency, but other translocations are extremely rare. Taken together with the GuideSeq data, this gives us confidence that we can successfully perform multiplex gene editing to generate fully allogeneic CAR-T product candidates with minimal risk of genotoxic or transformative events and a desirable safety profile. As discussed earlier, efficient and nearly complete knockout of the T-cell receptor is crucially important for a donor-derived CAR-T product that does not cause GvHD. Using Cas-CLOVER, we're able to remove T-cell receptor in up to 99% of resting T-cells. And to further increase the safety of the product, we're performing a purification step to remove any of the remaining TCR-positive cells, yielding a product that is up to 99.9% pure for cells with complete TCR knockout. Cells that are purified this way are not able to mount an alloreaction against donor-mismatched blood cells, as is shown here on the right in a mixed lymphocyte reaction type assay. While unedited T cells proliferate in response to the presence of donor-mismatched blood cells and dilute out the cell trace violet dye, Cas-CLOVER edited and purified cells are not able to recognize the donor-mismatched cells as non-self and do not proliferate. Using Cas-CLOVER multiplex gene editing in conjunction with our non-viral piggyback DNA modification system, we can successfully deliver our CAR transgene and edit resting T cells, thereby maintaining the desirable TSCM phenotype. Therefore, we're able to generate fully allogeneic, high-percentage TSCM CAR-T product candidates that confer the same product-like characteristics and tolerability profile that we have observed with our autologous P-BCMA-101 product. One significant hurdle that has been encountered in the allogeneic CAR-T field is that the genetic modification of the cells can lead to a functionally impaired cell product when compared to unedited CAR-T cells derived from a healthy donor. For our fully allogeneic, high-percentage TSCM CAR-T product candidates, we have been able to design a manufacturing process that yields a final product that functions just as well or even better than unedited healthy donor CAR-T cells. This is shown both in vitro by target cell killing, cytokine secretion, and high proliferative capacity, as well as in vivo mouse models of multiple myeloma, where we see comparable tumor control, as well as in vivo CAR-T expansion for both unedited healthy donor CAR-T cells, as well as for our fully allogeneic CAR-TSCM product candidate, P-BCMA-ALLO1. In summary, I hope I was able to show you today how we apply Cas-CLOVER gene editing to create fully allogeneic, high-percentage TSCM CAR-T product candidates. I've shown you that Cas-CLOVER gene editing can be performed in resting cells and is safe and efficient in knocking out the T-cell receptor to prevent GvHD and beta-2M to increase persistence of the CAR-T cells. Further, the high fidelity of Cas-CLOVER and its low to no off-target editing was demonstrated. Using Cas-CLOVER together with non-viral piggyback gene delivery allows us to generate an allogeneic CAR-T product rich in TSCM cells, which is predicted to eliminate alloreactivity as well as to maintain the beneficial characteristics of high-percentage TSCM products. Among these benefits are the correlation of best clinical responses with a high TSCM percentage, the tolerability profile due to gradual expansion of a TSCM product that could enable outpatient dosing, and the ability to give rise to potentially unlimited numbers of tumor-killing effector T cells that could be the key to CAR-T success in solid tumors. Taking together, we have generated an allogeneic CAR-TSCM product that is functionally equivalent to or even better than unedited healthy donor CAR-T cells both in vitro and in vivo.